Welcome to Unboxing Biology with Smriti. In the previous video, we have discussed about the Mendel's laws of inheritance. There were three laws: law of dominance, law of segregation, and law of independent assortment. In this video, we will be discussing about monohybrid, dihybrid, and trihybrid crosses. We will take some examples and solve this. So, mono means single, di means double. Okay, um, wait. So, di means double, and tri means triple or three. So, if we are taking here one character or trait and crossing between them, then it is known as monohybrid cross. And if we are taking two characters or traits and crossing between them, then it is dihybrid cross. Whereas if we are taking three traits or characters and crossing between them, then it is trihybrid cross. Okay, so we will start with monohybrid cross. Okay, now in monohybrid cross, we take only a single character and cross between them. Let us take example of a stem length. Stem length, okay? So the dominant form of the stem length is capital T, whereas the recessive form is this, small t, small t. Okay, so now we will do cross between this. We are crossing with the recessive form of the stem length. Fine. Now we are here taking the pure Mendelian's laws, which means we are taking here the pure lines of the parents. So here the parent is homozygous tall and homozygous recessive. Fine. Let us take this as a male and a female. And then we will, the next step is to form gametes. Fine. So these are the gametes which are formed by this parent, and the recessive forms of the gametes are formed by the female one. Okay, so they are gametes. After this step, we make Punnett square. Fine. So Punnett square for F1 generation. It means the crossing between the parents. Fine. So we have to make a Punnett square. Okay. Just make, because here the gametes are 2 in number, that's why we have to make just 2 columns and 2 rows. Now we will write here as a male and here as a female. Okay, now capital T, we will write the gametes over here, small t, small t. Fine. Now this is the F1 generation. F1 generation means, I already told you in the previous video that F1 generation is the crossing between the parents. So we are making here the Punnett square for the F1 generation. Now we will do the crossing. It will be capital T and small t, this and this. Now this and this, small t. Okay. So it will be again capital T, small t. Now caps T, small t, caps T, small t. Fine. Now, after making the F1 generation Punnett square, we will make or write phenotypic ratio of this Punnett square. Okay. Phenotypic ratio means the physical appearance. How? This is tall, this is tall, this is again tall, and this is tall. So the phenotypic ratio will be here 4:0. Where the 4 is tall and 0 is the dwarf. Fine. Now we will write genotypic ratio. Genotypic ratio will be the genetic appearance or the genetic makeup, you can say, of the possible genotype or the offspring. Okay, so it is genotypic ratio. Now the genotypic ratio will be here. This is capital T with small t, which means the dominant and the recessive both are present here. That's why it will be heterozygous in nature. So this is heterozygous, this is hetero, this is hetero, and this one is also hetero. So again the ratio will be 4:0, where 4 is the heterozygous and none is homozygous. Okay, now we will do selfing. I told you selfing means self-fertilization. Okay, let me write a selfing. Okay, so now we got the possible genotypes as capital T small t, that is the heterozygous tall. So now we will make here the cross. We will do the cross here between the offsprings. Now they will be acting like a parent over here. So in selfing, these will act as parents. Now we have to make gametes. Homozygous T, the dominant one and the recessive one. Here also it will be the same, the dominant one with the recessive one. These are the gametes. Fine. So just write here, these are the gametes. Now again, we have to establish here the Punnett square for this, and this will be now the F2 generation. Why F2 generation? Because here we are doing the crossing between the offsprings. Okay. Now just write this as a male. You can write over here, it is a male, and this one is the female. Okay, now just fill this. Capital T or the dominant one, the recessive one, again the dominant one with the recessive one. Now this is the F2 generation. Okay, so now we will do the crossing T with this T. So capital T, the dominant one. Now with this T with this T, capital T and small t. Then again capital T, this T with this small t, this T with this, so small t and small t, the recessive form. Now again, after making the Punnett square, we will be making or establishing the phenotypic ratio of this. Again, if you talk about phenotypic ratio, we are talking about the physical appearance. So it is tall. This one is also tall. This is tall, but this one is dwarf. So the ratio will be 3:1, where 3 will be tall in nature, where 1 is dwarf. Okay. Now the genotypic ratio. Genotypic ratio. Okay, so genotypic, this is homozygous tall. None of this is having the homogeneous. So the ratio will be 1:2. Now again, this is heterozygous tall, this is heterozygous tall, and this is not. Okay, so 2:2. The last one is homozygous dwarf. So again, this will be 1. So the ratio will be 1:2:1, where 1 is homozygous tall, second is heterozygous, or the 2 is heterozygous tall, and the third one is, that is this one, it is again homozygous dwarf. Okay, now this is the monohybrid cross. Okay, now we will do dihybrid cross. Okay, so dihybrid I already told you, which means we are taking here the 2 characters. Let us take the 2 characters. We can say like we are taking the stem length with the color of the flower, that is the purple or violet. Now Here again we are taking the pure lines, small d, small t, small p, the recessive form. Okay, now these are the parents. Again, the next process is to make gametes. So here the gametes will be 4 in number. How? Like this T with the P, this T again with the P. Now this T with this P and this T with this P. So we have 4 gametes. And the 4 will be same in number. Like, let me take different color. So it will be TP into 4. Fine. Now here it will be TP again into 4. So these are the gametes. Now the next step is to establish the Punnett square. Okay. So now here the gametes are 4. That's why we have to make 4 columns. Oh. Okay. So 1, 2, 3, and 4. I just made one extra to erase it. Fine. So now 3 and 4. We will just make a fast Punnett square, and this will be representing again the F1 generation. Why F1 generation? Because here we are crossing between the parents and making the Punnett square. Okay. So now this is representing F1. I'm sorry. This is F1 generation because here we are the crossing between the parents. Fine. So let us take this as a male and this is a female. Now the gametes are TP, the dominant forms of tall and violet, and the recessive form of dwarf and white. Okay, if this is T, if you want to write over here, you can also write it that we are taking the 2 characters, tall and violet. Here it will be dwarf and purple. Okay. Now just cross between them. It will be capital T, here small t, capital P, small p. And in all these 16, we have to fill the same thing because the same genotypes will be coming. You can see this T with this T, this P with this P. Okay, so all will be same. Fine. So again, after making the Punnett square, you have to make or you have to establish the phenotypic ratio. Okay, so here the phenotypic ratio will be 16 is to 0, where 16 is tall and purple, or you can say violet, whereas 0 is dwarf and white. Fine. Now we will establish the genotypic ratio. The genotypic ratio here will be, again, the ratio. This is heterozygous tall and heterozygous purple. So, again the ratio will be 16 to 0, where 16 is heterozygous tall and purple. I can say violet. Okay, whereas 0 is the other. Now we will just do the selfing thing. Selfing. Here we will cross between the offsprings that we got in F1 generation. Okay. Now here the gametes will be T, capital P, T, small t with capital P, and the small t with small p. Fine. Again here the gametes will be same. Capital T, capital P, the dominant and the recessive and the dominant, and again both are the recessive form. So we will get here 4 gametes. Just I will write over here that these are the parents and these will be the gametes. Okay, now after making the gametes, we have to make a Punnett square again, but this Punnett square will be for the F2 generation. Again, why F2 generation? Because here we are interbreeding between the offsprings. Fine. So we will make a first Punnett square. Okay, so now you will write it here as a male and a female. Now capital P with a dominant and a recessive, then recessive and the dominant and both recessive. Again here will be the same. Fine. Oh, this will be small t and capital P. This will be the small t and small p. Now this is the F2 generation. Okay, so now we will cross capital T, capital T, capital P, capital P. Now this will be capital T, capital T, capital P, small p. This will be capital T, small t, capital P, capital P. Now the dominant, the recessive, dominant, recessive, dominant, dominant, dominant and recessive, dominant, dominant, recessive, recessive, capital T, small t, capital P, small p, capital T, small t, small p and small p, capital T, small t, capital P, capital P. Again, we will just fill the Punnett square by just crossing between them. This will be a dominant and a recessive, recessive, dominant, recessive, dominant, recessive, recessive, recessive. So this one will be also the same, small t, and this will be the pure recessive. Okay, now we just have to make or establish, I already told you after making the Punnett square we do phenotypic ratio. Okay, so you can establish a fast phenotypic ratio. I will tell you the formula how. You just have wait. Okay, so you will do like this, establish 3 points. This is the easiest method by which, but this will only be followed, this rule will only be followed we are if we are using or doing the pure Mendelian's law, or we are following the pure Mendelian's law, that is we are taking the pure lines as the parents. Okay, so this you just have to join these lines. Okay, wait. Now, okay. Now with another color, I will be, you don't have to just make 3 points. Okay, 1, 2 and 3. Just join these 3 dots, make a triangle. Fine. Now with another color, just make 1. I guess I took the same color. So 1, 2 and 3. Just join these 3 dots, and the last one is this. Okay, so now you can easily establish the phenotypic ratio of the dihybrid cross in the F2 generation. How? You just have to write or see where these lines are passing. So 1, this blue line, 2, 3, 4, 5, oh, not this, sorry, this 5, 6, 7, 8 and 9. Fine. So let me erase this first, not this one. Okay, now these blue lines are passing from the 9 possible genotypes. Okay, so the first ratio is 9 ratio. Now the other one, so you don't have to in the second one you just have to see the dots. Where are these dots? So 1, 2 and 3. So you have to write here the 3. Again the ratio we will take the another color. So now again you have to see these three dots. So again it is 3 in number, and at last it will be the okay, so the last left is 1. Fine. Wait a minute. So this is 1. So the ratio of the dihybrid cross in F2 generation, the phenotypic ratio is 9:3:3:1. Fine. Now we have to write the genotypic ratio, but it will be a very lengthy process if we will count and then write the genotypic ratio. Okay. So now we will I will tell you one formula by which you can easily take out the genotypic ratio, but at the last. So leave this space. And now let us start with the third type, that is the trihybrid cross. Okay, so we will write over here the trihybrid cross. Now here we will be taking the 3 characters or traits and cross between them. For example, let us take the stem length with the flower color and the flower position. Okay, so axil position is crossed with the recessive form white, dwarf, white, and terminal. Okay, so this is the parents. These are the parents. Let us take this as a male and this is a female. Now we will take the gametes. The gametes here produced will be 8 in number. I will tell you. First, it will be TPA into 8. Fine. If we will see this T with this P with this A, then again this with this, and this with this. Now you have to do this and take out all the possible gametes. It will be 8 in number, and this will be these gametes. Okay. So TPA. Now here the gametes will be the recessive forms and into 8. Okay. So these are the gametes. Now you have to make Punnett square for all these gametes, but by making the Punnett square, it will be taking a long time. So I'm just telling you, the Punnett square will be same, and there will be 8 columns and 8 rows. You just have to write here male and female, and here it will be 8 in number, and here will also be 8. Okay. So now you just have to write capital T, P, and A. Here the small t, small p, and small a. If you will cross, it will be capital T, small t, capital P, small p, and capital A, small a. So these possible genotypes will be filled in all the 64 spaces. Okay, so now we have to write the phenotypic ratio, and this will be the F1 generation table. Okay, so phenotypic ratio will be here 64 because all of these will be same. So 64 is to 0. Fine. And the genotypic ratio will be heterozygous tall, heterozygous purple, and heterozygous axial. So again it will be 64 is to 0. So the phenotypic and genotypic ratio will be 64 is to 0 in the F1 generation in a trihybrid cross. Now the second step is selfing. I will tell you. Now the selfing thing. Okay, so here these are the parents, and we have to write the gametes. So here again the gametes will be 8 in number. There will be capital A, capital T, capital P, small a, capital T, small p, capital A, capital T, small p and small a. Now small t, capital P, capital A, small t, capital P and small a, small t, small p and capital A, and at last it will be all the recessive forms. So now these are the 8 gametes which will be produced by these parents. And with this thing also, we will get all the same 8 gametes. Fine. Now again, we will establish the Punnett square. Fine. And we will get all the 64 possible genotypes in the F2 generation. Fine. So I'm again telling you like this. T, P, A, and you have to write all these 8 gametes. These 8 gametes over here, these 8 gametes over here. So now it will be a very lengthy process to write. So I will tell you again the formula to obtain the phenotypic ratio and the genotypic ratio of the F2 generation in the trihybrid cross. Okay, so now we will study the formula, how to calculate it. So for the phenotypic ratio, we have a formula 3:1 to the power n. Okay, so n is the number of traits or characters. Okay, or characters you can say you are taking. So for the monohybrid cross, in the F2 generation, this will be applicable. Okay, so 3:1 to the power n will be 3:1. So for the monohybrid cross, the phenotypic ratio will be 3:1. For dihybrid cross, it will be 3:1 to the power 2, because 2 characters are being taken for the cross. So how we will calculate it? 3:1 into 3:1. The formula to calculate is multiply this 3 with first this and then this. Now you have to multiply this 1 with 3 and then 1. Okay, so 3 into 3, 9 is 2, 3 into 1, 3, then again 1 into 3, that is 3, and 1 into 1 is again 1. So the ratio which we will be getting, the phenotypic ratio for a dihybrid cross, will be 9:3:3:1. Fine. Now in the trihybrid cross, we are doing here 3:1 to the power 3. Okay, because 3 characters are being taken and crossed between them in the trihybrid cross. So 3:1 into 3:1 into 3:1. Okay, now we have already calculated this one in the dihybrid cross. So we will directly write over here 9:3:3:1 into 3:1. So again, it will be same thing. This 3 with all the 4 numbers and 1 with all 3 numbers, 4 numbers will be multiplied. So the answer will be 3 into 9, 27, 3 into 3, 9, 3 into 3 again 9, 3 into 1 will be 3. Now 1 into 9, 9, 1 into 3, 3, 1 into 3, 3, and 1 into 1, 1. Let us rearrange it. So 27 is 2, 3 times 9, and 3 times 3, and then 1. So this is the phenotypic ratio in a trihybrid cross. Okay, now we will study for the genotypic ratio, how to calculate it. So the formula is 1:2:1 to the power n, where n is again the number of trait or the characters being taken. Sorry. So first one is monohybrid cross. Again, it will be 1:2:1 to the power 1, which will be same 1:2:1. In a dihybrid cross, it will be 1:2:1 to the power 2, which will be 1:2:1 multiplied by 1:2:1. So again, the formula of multiplying it is the same. This 1 with all the 3 numbers, this 2 with all the 3 numbers is multiplied, and again this 1 with all the 3 numbers. So the dihybrid cross will get the genotypic ratio as 1 is 2. 1 into 2, 2. 1 into 1, 1. 2 into 1, 2. 2 into 2, 4. 2 into 1, 2. 1 into 1, 1. 1 into 2, 2. And 1 into 1, 1. Fine. So this is the genotypic ratio which will be obtained in the dihybrid cross. Now, the trihybrid cross will have 1 is to 2 is to 1 to the power 3. That is, we have to multiply 1 is to 2 is to 1 into 1 is to 2 is to 1. Fine. So we have already calculated this in the dihybrid cross. So we will write over here 1 is to 2 is to 1 into 2 is to 4 is to 8 is to 1 is to 2 is to 1. Fine. Into 1 is to 2 is to 1. So now 1 with all the numbers will be multiplied, with 2 again all the numbers, and with 1 again all the numbers. So let us do it fastly. 1 with when we will multiply it with 1, so all the number will be the same. 4 is to 2 is to 1 is to 2 is to 1. With 2, 2 ones are 2, 2 twos are 4, 2 ones are 2, 2 twos are 4, 2 fours are 8, 2 twos are 4, 2 ones are 2, 2 twos are 4, and 2 into 1, 2. Now again, 1 will be multiplied with all the numbers. 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So this is the genotypic ratio which will be obtained in the trihybrid cross. This formula is only been applicated when we are starting with the pure lines or the pure Mendel's laws. Okay. So now in the next video, we will be discussing about the test cross. Okay. So thank you for watching my video.